Good morning to everybody. I thank you a lot for being here. I thank Mark for having this wonderful forum for exchanging ideas between scientists, clinicians, patients. I thank Board of Directors and also Scientific Advisory Board, and my hat off to Dr. Preceding as a frontier in the fight against mesothelioma. Surely what Isaac Newton said about the reason that he can see far is that he sits on the shoulders of giants is very much applicable to Dr. Preceding and the rest of the generation of scientists who went very far because people like him made the field clear and made the path paved so we could continue. So I'm very glad and very honored to speak at this forum today. So basically what I want to bring to your attention is a bit of a work that we have done on this topic named cancer stem cells. And why is that important and why is that exciting in treatment of cancer in general and also targeting mesothelioma specifically? Well, we call it Achilles heel of tumors basically for the reason that during the last 10-15 years we have come to understanding that not all tumor cells are the same and some of them are much more important than the others. I don't think I need to explain about mesothelioma to you. That would be preaching to the choir. You all know about how the subcategories and also the clinical ramifications and things like that. But in my lab, what we are trying to do is that we are trying to develop therapies which go after stem cells generating this deadly cancer, either cancer stem cells from mesothelioma origin. Now, for us to have a bit of a warm-up to the idea of cancer stem cells, I just want you to imagine a war scenario in a battlefield. You have generals and then you have soldiers who carry on the orders. So one of the ways that you could defeat an army, which, again, using an example of war, I don't think it's a really happy example, but it works the best here, is just to try to go and kill every soldier and every member of the opposing army. The other way would be if you could take out the generals, if you could take out the commanding officers, and that would completely disrupt the functionality of the opposing army, and the foot-ground soldiers are not going to be able to carry on, and you essentially defeat the opposing army. So cancer stem cells, indeed, in context of a tumor, they are the generals. They, in resemblance to what normal stem cells do in a tissue, they basically raise the whole different types of cells which are needed to populate a tumor. And most likely, one of the reasons that to this date, in the year 2014, we are standing here and still having a significant need of novel therapeutics for cancer therapy in general and for mesothelioma treatment specifically is the fact that we have been trying to target foot soldiers, what we call differentiated cancer cells, and the cancer stem cells, the generals, have been hidden from us. And it doesn't matter, you can kill as many as differentiated cancer cells as you want, as long as these engines, these queen bees exist there, they simply repopulate the tumor back. And if we could come to an understanding of how to target the engine behind the tumor repopulation, the ones who matter, then we must see dramatic change. And luckily for us and also our future generation, the thought is moving that way, and we are coming to an era that more and more of cancer stem cell targeting agents are coming to the attention, and we can see very promising and impressive data from this. This is the picture of, this is a statue of Achilles in Achilles Garden, and also for the topic of our discussion, this Achilles heel is going to look like something like this. So we're going to take a look at the 20 seconds animation, if you could go ahead with it, and then I'll explain simply on it, and then we're going to go take a look at some of the more interesting data. And this is an animation about cancer stem cells that they put together at Cancer Research Foundation of UK, but I thought that it would be useful. So basically, it doesn't have voice. I will just narrate it for you. It says that tissues in our body are, the normal tissues are composed of a number of cells, and some of them have the stem cell characteristics, meaning that basically what happens is that the stem cell not only produces its own kind, but also produces the other kinds that are needed for formation of a normal tissue, and later on for an organ. So what happens is that moving? What happens is that basically a stem cell, this is supposed to something happen, here we go. A stem cell divides, it produces its own kind, also it produces the foot soldiers, the differentiated cells which form the tissue. Now, that is good and that's jolly and that there is no problem, but once a stem cell starts acting weird, it starts producing cells that are not normal and itself is not normal, that's when the problem comes in, meaning that you have a scenario where the stem cell is mutated to a not healthy version of itself named cancerous stem cells, and now cancerous stem cells develops other cancer cells which are not normal, they don't carry on with the regular functions such as differentiated mesothelioma cells, and they also develop other cancerous stem cells populating the tissue with wrong kind of cells. And eventually you have a tissue formation which you have a bunch of cancerous stem cells and a bunch of differentiated cells. If you kill the regular cancer cells, the cancerous stem cells are still there. So all they do is that they simply just repopulate the tumor back. And that's what we have seen in the last maybe 40, 50 years. I mean, not that bad. We have come way far with our treatment in oncology, but in many cases you see a relapse coming back. You know, at some point, unfortunately, the bad news is that, can we go back to the presentation, that the tumor is coming back. So the theory was very attractive. Calling cancer stem cells a theory probably is a little bit of injustice right now, but it has been, you know, like any other new ideas in the field of medicine, it always faces resistance and criticism. But I think during the last 2 years especially, significant level of data came out to the support of this understanding. And now we are looking towards drugs which can target these cancer stem cells. Okay, so one more time. The most important message so far is that if the yellow is a cancerous stem cell, if you take that out, your differentiated cells are going to be there, they're going to hang around for a while, they're not going to be able to cause a significant tumor, they are not going to be able to have significant level of invasion, metastasis, causing a lot of clinical issues later, and they basically run their time out. If you can't do that, if you leave the cancerous stem cells, the game is back, full potential cancer, after a period of delay. And I'm going to show you some experimental data that when we targeted cancerous stem cells in mouse model, a significant level of lack of tumorigenicity in vivo and in vitro happened. So again, the slides basically tell you the same thing. A successful therapy, one more time, here is the ones that takes cancerous stem cells out. Then you will end up with some sort of what they call maybe benign tumor or maybe a tumor formation which is, you know, from biological point of view is not, so to speak, malignant and can be dealt with easily. But if you leave the cancerous stem cells in gray here, you just have the tumor relapse. There are multiple theories about where cancerous stem cells come from. They are mutated form of normal stem cells, one theory. No, your regular cancer cells de-differentiate. They take a step back in differentiation. That's actually very interesting. And they become cancerous stem cells, meaning that they give up some of their expertise to become queen bees. They basically don't act as a differentiated cell, and they go back and they become pluripotential cells. But not to get that much tangled up with the theories, there is one relatively dominant idea in the field, which is called tumor hierarchy model, which says at any point, the tumor is designed in a way that it puts the flagship of stem cell characteristics on a minority cell population. It's just like an army that has the capacity to say, okay, you are my commander right now, and 2 hours later, no, you are not the commander, you are the commander right now. If that was the case, it would be really hard to take out that commander because it switches them so fast. And that's relatively a scary theory, and I'm hoping that those guys are not that correct, but doesn't matter who is wrong, right and who is wrong. We are developing tools to target many different scenarios now. So the idea is to take out the stem cells of the tumor. How can we distinguish a cancerous stem cell from other population? There are a number of markers, either on the surface of the cell or inside the cell, that to this date we think that they dictate the stemness. Are these markers, all of them, completely cancer specific? Probably not. But a lot of them, you see enhanced level of them in tumors, tumor cells and tumor tissues. Do we always match them with the staging of the malignancies, i.e. people with more advanced levels of malignancy would have more of these markers? Usually yes, but then again, nothing is completely black and white in research. Things can be a little bit here or there. So of all of these markers that we have researched, one of them which is dominant is CD133, which was protein of the year in 2007, something like that, was a content of conversation, sometimes very emotional for a long period of time. But again, I think evidence is significantly overwhelming that in the context of many tumors, CD133 can tell you where to put your money down in terms of cancerous stem cells. It's also a topic in mesothelioma. There has been a number of reports of CD133 expression in mesothelioma, and part of work goes on to see what other markers are there for mesothelioma. When I took a look at the field, these are a list of some of the markers that are prominent for knowing which parts of mesothelioma cells are cancerous stem cells. And these have been developed with different strategies. One interesting thing is that one of the ways you can understand what markers are for cancerous stem cells is that you just add chemo to the culture of mesothelioma cells, and then the ones who die off, never mind. The ones who survive, you look at their markers, and they're usually enriched in certain markers, meaning that they can survive chemo because they are cancerous stem cells, and you pick on those markers as the markers for cancerous stem cells. I believe my other colleagues have given talks. I think Dr. Varghese spoke about it, and also a company, Veristem, is also looking into targeting cancerous stem cells, which I believe they will talk later on today. Three markers here, CD133, as I spoke to you, a marker named EZH2, and another marker, VEGFR2, have been under investigation in my lab as a way that we could launch an attack against cancerous stem cells. This is a picture of CD133. It's just a protein on the surface of the cells, and don't want to get too technical with that. So now, how can we target them? Drug therapy is always a possibility, and gene therapy, and from the family of gene therapy to the expertise of my lab, oncolytic viruses. Now, again, when you are in some sort of business, I guess you have to kind of, advertise that a little bit. Now, everything is very cool with drug therapy. The advantage of it is that you simply have a possibility of improved pharmacokinetics, and designing drugs against certain targets is something that we have come way too far, and now we can move very fast. It used to take years that you could design a molecule. Now the time has shortened and promising things come out. But one of the problems of drug therapy in general has been development of resistance. And again, we have heard about that repeatedly, that when a cancer cell feels that one of its pathways, one of the ways for its survival is under attack, it simply shuttles back its biology from that way of life to another way of life. Cancer cell is a very versatile, very resourceful being. There are lessons in that to be learned. When we are talking about the gene therapy, especially attacking cancer cells with a virus, this attack comes so strong that if we could direct it to the right way, if we could have it powerful enough, the cell is not going to have enough time to shuttle itself to another way of life. A virus, such as the viruses that we work, herpes simplex virus, a natural virus in 16 to 24 hours kills the cell. The cell, viruses don't take hostage. It's not possible to escape from it. But then, the negative things of working with viruses is that, well, you're injecting somebody with a virus, and number one. Number two is that you have to be able to control it. The virus has be able to act specifically against cancer. This is the promise of field of oncolytic viruses. Viruses against cancer which target cancer cells specifically, and they leave normal cells untouched. It's not more than 15 years. It's not a field that has been going on for a long time, but the idea of viruses being anti-cancer agents goes back to early 1900s. Doctors would see that patients who had, for example, lymphoma, during a period of viral infection, their tumor was regressing really good, and then when the viral infection was cleared, the tumor would come back and kill the patient. It was giving them the idea that the virus has some oncolytic effects. When a virus attacks a cancer cell, it releases two series of antigens, which not only virus directly kills the cell, but release of these antigens triggers host immunity, anti-tumoral response from our body. One series are viral antigens that are released, and the other series of antigens are tumor-associated antigens. So these anti-cancer viruses not only directly kills the cancer cells themselves, but also call the air attack, being our immunity to act against the tumors. Now, the slide tells you the same thing. Once you target a cell with these viruses, a number of other mediators are released that they boost your anti-tumoral immunity. Now, these two slides, which you can't read, and neither can I, tells you the list of, you're not supposed to be able to read it. I'm just supposed to impress you by what a long list it is. It gives you the list of viruses which are in clinical trials. So right now to this slide, you might have thought that, okay, this is another thing which, this guy is talking about and may or may not happen, and you may not ever hear about this again. But no, that's the wrong impression. To this date, there are 15 members of oncolytic viruses in clinical trials. There are 7 or 8 members passing phase 2 and phase 3 clinical trial. There is one version of oncolytic viruses in China as an approved drug given to more than 10,000 people against head and neck. And if you go on their website, I believe they are advancing that virus to be tried in a number of other malignancies, and maybe mesothelioma would be on their list. Actually, I wanted to get in touch with them to see what are their plans against targeting mesothelioma with this. We have the virus which my friend Dr. Kratsky works on, a mesovirus against being tried right now in recruiting patients and studying against mesothelioma, and we are entering the phase that more and more number of clinical trials are going to open up for mesothelioma with these viruses. This list is a more simplistic list of what is in clinical trial. One significant advancement to the field was when in 2000, that says 2011, but I think it was 2012, Amgen paid $1 billion to buy one of these viruses from one of our very lucky colleagues or coalition of colleagues. It was a mutated version of herpes, the same kind of virus that we work on in our lab, and now it's clearing phase 3 in targeting melanoma. So good news on the side of the viruses are coming. These traditional enemies of us are now coming to our help with the help of genetic engineering, with the help of molecular biology, and with the support of entities like MARF that put down the first $50,000, the first $25,000 that a young scientist somewhere needs to just get this idea going. So all these now high glamour and very fantastic ideas which eventually, big agencies jump on, they have to start from a certain seed money, and places like MARF, especially MARF in mesothelioma, is the one that injects the first, blood to the vein of these projects. So, okay, now giving you a little bit of a tour of what we do in my lab. So we spoke about cancer stem cells and we spoke about viruses. What if we merge these two ideas? Number one thing that we remember is that you don't need to kill each and every cell in a tumor. Just go after cancer stem cells. Number two, you got a virus. So design it in a way that it only goes and kills cancerous stem cells. And that's what we basically put forward here. There is a minority fraction of cells which are cancerous stem cells. If you had a virus which would go and take them out, they would leave, basically. They would be infected and killed. There are some differentiated, regular cancer cells left. They would run their lifetime, but there is nothing to back them up. These things cannot regenerate themselves forever. That's the job of the cancerous stem cells. Eventually, the whole house of cards is going to come down. And let's see if we could show you evidence that that actually exists in reality. Okay, we designed this virus which targets CD133. I told you it's an important cancer stem cell marker. And the virus replicates and destroys CD133 positive cells. It doesn't go after CD133 negative cells. Then what we did is that we mixed the virus with cancer cells. None of this is mesothelioma. We are working on mesothelioma right now, and I will show you some mesothelioma-related data in the next few slides. But we basically mixed them with cancer cells. We injected them under the skin of the nude mice, a mouse that growth of human tumor cells is possible in it. So the mouse immunity should be really, really low or nothing. Otherwise, if the mouse is fully immune, it's going to clear human cells. So if you mix this in 3 different models, we mixed it, we injected it subcutaneous, almost no tumor growth, nothing in any of them. When the virus was present, it was impossible for the cancer cells to grow. And you can see that the right side of the leg is where we injected the cancer cells, and the right side we injected the cancer cells, same numbers with the virus. Huge tumors on the left side, which is the control, almost nothing on the right side. So this mostly tells you about the fact that if the virus comes at the site of the tumor, if it arrives at the site of the tumor, it will block further tumor progression. It's mostly a preventive matter right now. What about if the virus is already developed? If the tumor is already developed, and now we attack it with the virus? Can that cause regression with a pre-established tumor? The answer is still yes. We injected the cancer cells subcutaneously. We let them grow to about 100, 150 cubic millimeter, which is kind of actually not a bad size compared to the size of a mouse. And then we just injected them intratumorally with 5 injections of our virus. And you can appreciate a good drop in the size of the tumor growth compared to the control side. Only 5 injection 3 million virus. So we are just simply continuing this. We are adding to the number of injections, and we are also experimenting to let the tumor even grow bigger and see if we could target it. So that was a virus against the marker CD133. We also have developed a virus against a very interesting marker which is out there recently, and there is no specific blocker for it as we speak right now, named EZH2. EZH2 is a protein which when it's overexpressed, it simply goes after our good anti-tumor genes and suffocates them, simply. So by suffocating our good anti-tumor genes, it leaves the field for the oncogenes, or relatively not so friendly genes, to take over and basically cause neoplasia and later malignancy. So we have developed the same story, and this is the data about mesothelioma. LP9s are relatively normal mesothelial cells. Everything else in this slide, majority of them are mesothelioma cells. And you can see, just look at this slide, that LP9 does not have this EZH2 protein expressed that much, at least below detectable level here. And the mesothelioma cell lines express a lot of it. And other people have looked at it. There is not a lot of work in EZH2 field, but there is a few papers out there. In tissues is the same. So we went and designed the virus against it. Same story. The virus goes inside the cell. If it's EZH2 positive, it replicates, blows it up. And we are now doing in vitro phase to see, first of all, in vitro, in tissue culture, is the virus going to kill the mesothelioma cells? The answer is yes, and very strong. Is this virus going after cancer stem cells which are positive for this marker? Is something that we are now pursuing. Last 2-3 minutes, not to go over time. One level of cooperation we are doing with UPenn is that, okay, now my virus is a virus which goes inside the cells. If the marker is positive, replicates, blows up the cell. If the marker is not positive, the virus is going to stay dormant, and systems inside the cell are going to chop it and kill it apart. Okay, but if you injected IV, it goes everywhere in the body, right? What if we coated this virus in a way that it would only get trapped and get concentrated inside the tumor site and does not go anywhere else? For example, if you have a tumor in site A or B, if you have mesothelioma, if you have other types of malignancies, the virus only gets trapped and gets concentrated at the tumor site. So 2 levels of specificity. Virus only gets concentrated at the tumor site, and once it's there, it replicates only in cancer cells. We are working on that with the help of these new generation of DNA antibodies called aptamers, which are just like antibodies, but the structure is actually nucleic acid, and they are proven to be very specific and very strong for any marker that you develop them against. We have now coated one version of our virus with a nanocoat, basically circling the virus, decorated with these aptamer molecules. And these aptamer molecules can be designed against different malignancies such as mesothelioma. There is some work on mesothelioma. We are enhancing that work to develop aptamer specific for mesothelioma. And what we predict is a model like this, that the virus is coated with this aptamer, goes to the site of the tumor, goes after cancerous stem cells, destroys them. Later on, the whole tumor gets disseminated. But inside a tumor microvasculature, this scenario happens. You also have leaky endothelial cells, which makes your virus be able to penetrate inside the tumor. But in a normal scenario, where you're in your normal organs that you don't want to harm, the scenario is different. Your virus basically does not have enough affinity to bind because of this coating that you have. And on the other side, the endothelial cells are relatively nicely attached. So enhancing the specificity and therefore reducing the side effects of this form of therapy. The first version of the virus, which was given to us, it was nano-coated, sent back to us. We just wanted to make sure we didn't kill the virus by this technology. And we just checked to see if the nano-coated virus is still infectious, and that seems to be true. Ultimately, I mean, I don't know if, probably within next 5-6 years. The idea is this, that somebody walks into clinic and has mesothelioma or has cancer A or B. We take a sample. In about a week, we determine what are good markers for this person individually, which are significantly high in the tumor and are not present or very low in the normal cells. And in about 2 months, with my lab technology, right now is 2.5 months, we can develop the virus for that marker for that person. If the money and support and intelligence and everything existed, I'm sure this period of time can be cut from 2 to 3 months, probably to about 15-20 days. We can develop this designer virus for that person and immediately give it back to him. One level of testing could be that the virus could be tested in patient-derived xenograft model, where you have a piece of that tumor growing in mice from that patient. We tested it in that to make sure it's working, and then could be systemically administered to the same patient. This part, kind of like designer virus for each patient, is a little bit futuristic, but so was many drugs that are now on the shelves of the pharmacies 20 years ago. There is no reason not to dream about a good cause. So that's about it. At the end of the day, just hold on where you are. The help is on the way. Thank you.